So welcome here, everyone. I'm Erik Söderbäck. I work for Covaris, and we will talk about FFPE in oncology settings today. So we have an agenda. I will go through what Covaris can provide in terms of sample preparation and also library preparation. We will do an introduction. So Nadia will do an introduction from Illumina. And then we'll show a collaborative effort from Covaris and Illumina where we optimize the workflow for phi-base, methylation, and mutation sequencing. And it's a streamlined solution for FFPE analysis. And then we'll end up with Professor György Markó-Varga from Lund talking about the proteomics workflow from FFPE material. So he will describe then how to prepare protein and in a mass spectrometry workflow. So Covaris, probably most of you know Covaris as the DNA shearing company, but the technology we base our solutions on is called Adaptive Focused Acoustics, and that can be used for much more than just the shearing of DNA. So it's a disruption, so it can be used for disruption, cell lysis, molecular processing, and the trick is that we transfer very accurately the energy to the sample. And it's a non-contact isothermal process which eliminates any cross-contamination and it preserves the analyte integrity as well. And for this we have the consumables, accessories and reagents that are optimized for the application of interest. And we can say that you can work with more or less any kind of sample types and with any kind of biological molecule. So our offerings can be summarized like this, that we have instruments from single sample handling up to high throughput, 96 and 384 well systems. And for this we have then the consumables and the reagents that are required for certain applications. And we can automate this as well. And I'll come back to the automation regarding FFPE. And for this, we serve more or less any market that uses biological molecules. And we can also support applications for any kind of omics. So we have solutions for genomics, transcriptomics, methylomics, or methylation analysis. We can also do for metabolomics. But going into the true extract, which is our reagents then for extraction of DNA and RNA from FFPE, this is quite a simple workflow, and we use the ultrasonication device then to emulsify paraffin, which is a big difference then from other vendors that have organic solvents for the deparaffinization. So in the step here, we use the alpha technology and we emulsify the paraffin, which gets small pieces of the paraffin and the tissue out in an aqueous buffer. And by that, we can efficiate the proteinase K treatment downstream. We spin down the sample, which makes the DNA ending up in the pellet and the RNA in the supernatant. And by purification of both these fractions, then we have DNA and RNA from the same sample, which can be used for further downstream processing. And the quality is very good, both for DNA and RNA, and particularly the RNA sticks out as very high quality with a high molecular weight RNA. But to show that also the DNA is of high quality, I want to emphasize an application note recently from PacBio, which show that with DNA from FFPE, you can do long-range sequencing. And with amplification library preparation, you can reach very long reads then. And they claim that these results demonstrate that even with degraded FFPE material, it is possible to recover DNA fragments of sufficient quality and length to enable high-fi long-read sequencing. For short-read sequencing, Covaris has during the last year introduced library preparation solutions for whole genome sequencing as well as for RNA sequencing. These can be used for any material, but we have optimized workflows for FFPE DNA as well as from FFPE RNA. And now, latest here, we have also the multi-omic library prep with Illumina 5 base, and that's what we're going to focus on now. So I leave this to Nadia.
Thank you so much, Erik, for the opportunity to tell you a little bit more about the 5-base solution and also our collaboration with Covaris. We've been partnering already for a long time, also from the time of the TSO 500 assay, and it's very nice to see that we're taking it also to the next level with the expanding range of applications. So, and we jointly, we believe in the power of the multi-omics approach as the way to reveal, more complex biology and to see the fuller landscape of what, and to ask the questions that the cancer biology really deserves. So with this in mind, we've been from Illumina side active on the sides of the very different omes. Nowadays, many of them can be addressed with the next generation sequencing. And thinking about DNA in general, it's, it's in the DNA's biology to be a multi-omic molecule because it brings two levels of information at the level of genomics, so with all the variants that one might be interested in, but also the additional level of regulation then epigenetics brings in particular methylation. And this is exactly what we are trying to address with the Illumina 5 base solution. The innovation bit that we are bringing here, Illumina is earlier, just it's been still kind of a recent announcement from the last year. We brought it to the market. This novel type of the library preparation that allows you in one go, in one workflow, without the need of the splitting things in two, get the information both from the genome level, right, in terms of when you're looking for the variants, but also at the same time getting the full picture of the methylation profile. And, I would not go into too many details, but the essential thing, how it works, the first step is bringing the innovation on the side of the chemistry. So on the left side here, you see the traditional NGS-based methods or bisulfite sequencing, when you would need to treat the molecule of interest with the enzymes doing the non-direct conversion. So everything that had the methylation will be sort of protected, and then, all the non-methylated Cs would be converted into Ts. As a result of this, the methylation component was there, but the library complexity was quite, shifted, because essentially you are ending up with the overrepresentation of Ts. And also this has been quite a harsh treatment that does not preserve the integrity of the molecule. So the approach that Illumina take in bringing their solution to life is really modifying the way that the chemistry works. And this is what we are looking at the right side of the slide. Illumina 5-based conversion chemistry enables this direct conversion of the methylated Cs into Ts, and that brings a whole number of advantages that I'm going to highlight just on the next slide. So by doing so, we're essentially addressing the gaps in traditional methylation sequencing methodologies. As I mentioned in the beginning, the assay is made to bring you the dual insights, so the high accuracy detection of both methylation and genomic variants. It comes with the advantage of the seamless workflow, and again, with the partnership with Covaris, we can really take it from the step of the extraction all the way to the data output, and it drives the cost down significantly. I would kind of, like to also give you a snapshot about the workflow, what it takes from the kind of, library preparation into getting the insights. So the library preparation step takes less than 1 day. And then we are speaking about, the sequencing part that is also can be done on the range of Illumina instruments, and we are wrapping this part up with a very important bit. It's the power of the software, because again, as I always like to quote, it's software that brings the data, that makes the data the insight. So this is the overview how it works, and the data analysis part is enabled by the Dragon pipeline being the part of this integrated solution, so you don't really need to bring the additional bits and pieces. And you know, as our collaboration with Covaris, we started, you know, we started sort of with the first testing on the workhorse, right, of our customers with the LE 220, but then we expanded basically this workflow, the testing of the workflow, to the other platforms as well. And Erik, I will invite you to stand next to me to comment on the outcome.
Sure, the instruments you see here, so the workhorse called here, the LE220 is present in a lot of labs around the world. But during the last years, we've introduced a high throughput solution called R230 and also one medium throughput ML230, which both works in 8 strips, but also the R230 can work in 96 well format. And it can also be automated, and I'll come back to that. But in principle, what we showed here with genomic DNA, the Covaris, the instrument, supports the 5-base workflows, irrespective of the throughput then. So we have scalable solutions then for the 5-base. And the Illumina 5-base and Covaris then consistently deliver a high confidence variant calling and methylation profiling. This was from genomic DNA, and then we took the next step then to optimize also workflow for FFPE. So here we took the genomic DNA from a reference DNA, and we reshared that down to 200 base pairs to have it equivalent to what we get from the FFPE, and we compare the outcome then from the library preparation and sequencing. And just looking what the cell cultures that are then formalin-fixed and embedded in paraffin, and we extract that with our TrueExtract solution and run through the 5-base workflow. You can see here it's very consistent then with the library traces you see to the left, and also the amount of library that we get out from this. So irrespective if it's genomic DNA or FFPE DNA, we can provide the workflows. And also the QC metrics from the sequencing shows very robust data from the FFPE as well. So here it shows that we get more than 200 base pair on average insert size, and it's no impact on mapping or the methylation analysis. It's equivalent between the genomic DNA and FFPE DNA. We also investigated the variant calling from this, so both the SNPs, single nucleotide variants, and indels show very high consistency between the genomic DNA and the FFPE DNA. So from this we took it to also, of course, to look at real patient samples. So the last step then, we took lung cancer and compared to lung normal tissue. And we extracted this with TrueExtract and run it through our R230 platform for both the extraction and the library the DNA shearing and library preparation. And again, so to the left, you can see the size distribution then of intact, the FFPE DNA, which is very intact then. And it's quite high. It's distorted then with the x-axis here, but the peak range here is about 5,000 base pairs from FFPE DNA. And when we share this for the optimized workflow for the library preparation with 5-base, it's about 200 base pair, and that's the trace you see to the right, that you get very consistent library, the fragment size for the library preparation. And to sequence this, again, we show that the quality metrics are very equivalent here. So for both the lung cancer tissue and the normal tissue, we show consistency for the coverage, the mapping, but also the insert size around 200 base pairs. So to summarize the results from this, we show that both we have a scalable solution for the instruments up to very high throughput. We can show consistency with the methylation we show here. It's consistent with the positive and the negative controls. both input-wise, so we compared here 50 nanogram and 100 nanogram. We compare genomic DNA with FFPE DNA, and the results are very consistent both between genomic DNA and FFPE DNA. So to summarize this, I give Nadia the way to describe it from the Illumina standpoint.
Thank you so much. So today what we tried to show you is also the way how, together with Covaris, we did really go in a nice direction in streamlining the genomic and methylation profiling for FFP material, providing this comprehensive solution that really can allow you to do more samples, to do them faster and then accelerate the research workflow, starting all the way from the nucleic acid extraction, also with the true extract prep from Covaris, going into the DNA sharing that is here across different platforms, so depending also on the throughput of your team and the accessibility of the instrument. This is a really great point. And then seamlessly going into the library preparation, one library prep, so no additional kind of workflows, sequencing as an outcome, you get the information two-in-one, you get the information about the DNA sequence and the full methylation profiling. So this is exciting. We've been now partnering, of course, on establishing, testing these methods, and we are working on the preparation of the joint technical note, so this will be also available very soon together with the other resources that are out there already. Thank you so much.
Thank you, Nadia. So with that said, we'll stop this and we go over to Professor György, Markó-Varga from Lund, to describe a proteomics workflow from FFP as well.
Okay, good afternoon, everyone. Very nice to be here. I'm György Markó-Varga, and I'm going to tell you a little bit about the development that we have with formalin-fixed material. So this is a great asset since pathology department have very, very big collections over decades with these samples. So I'm going to show you some studies on melanoma, and you're well aware, I mean, this is the one of the most fast progressing metastases, cancer disease of all. And we also have a very high mortality rate that actually we have not really been able to conquer and don't have a very good solution for, even though we have new pipeline for big pharma introducing immunotherapy and other ways of handling it. So here is a little bit of statistics of where we're going. Still going up, even though diagnosis is not the best, and treatment is also, we would like to expect more. Now, that is true both for men and women. You can see here, top scale, you have Australia and New Zealand, and below you have countries like UK and Scandinavia actually is pretty bad. However, if you find the diagnosis very early, you have a very good chance. And as you move into, let's say, stage 2 or so, not so good. So because then you're already under pressure. And then we are just, in principle, in the hospital, we're kind of extending lifespan of patients. But doing so, we have a clinical team. We really work together, multi-center studies around the world. So here's Olivia Esto, Marcel, and Balázs. And also, we work a lot into the clinic as well in surgery. And in surgery, we have a very good established collaboration. So we actually get clinical material. So what you see here is in a hospital where we are just down the corridor and able to process and handle surgically isolated material. So from there, of course, then you have the workflow in the hospital. You go into pathology. And now we built a super big biobank. It's actually here in Hungary in Central Europe. And to the right-hand side on the image, you see where we already started. So this is 650,000 tumor blocks that we enter in. And in the middle, that's actually the whole archive with 6 million. And to the left-hand side, you see how everyday work is done and how they are cataloged. Now, we are addressing actually a build of knowledge in understanding both intra-tumor heterogeneity and inter-tumor heterogeneity. So we are interested to understand, I'll show you a little bit later how we do that, the clone specificity. So how can we identify that? And what about the immune defense? How does that work out together? And we are now moving up to very high numbers of images and patient tumors where we are kind of building knowledge on that. And in the end of the day, of course, many of us in my team is from big pharma originally, now moved over to hospitals and into academia as well. But we are very interested in the mode of drug action and also to understand the mechanisms driving that. But in the end of the day, the most important is actually to go where the problem is, and that is within the tumor tissue. So here is an example. Early on, we started to catalog. We built very big databases where we started to annotate these different regions. So this is like early setting, and if you go from there, you can actually now start to do modeling. So you can find different compartments within the tumor where you can identify different tumor clones. And if you go one step further, you can actually then move from there to have single cell annotation. And in this way, you will have very high numbers. So imagine you get like 600,000 cells identified within one section. And then you move up to 1,000, 10,000, 100,000 of sections from multiple thousand patients. Well, you get my point. And then from there also we are kind of attacking in a single cell mode. So on the right-hand side, you can see how we isolated single cells within tissue from activity that we're driving by laser microdissection. And the whole idea here is also to have spatial readout and to understand what is really happening in compartments within the tissue. Now, building this up, we decided, so Letizia and Lucas have been pushing this now for some time, to use an open source, so it's a QPath, it's available publicly, and then we kind of build our model platform on this platform. So you start with setting it up, getting holistic whole section view, and then as you move forward, you're able to move into the details and then also to build neural network, and from the neural network you're actually able to get to a situation where you have an ability for automated annotation, and that is on single cell level. So, you know, there are not so many pathologists in the world that will be able to help you, so we need computation power in order to manage go forward. And so what we're doing here is that we're using augmentation technology, and I show you a little bit of an example here. So what do? So you have one original image. This is the one to the left, right? And then what you do, you actually make a virtual copy of that. What you do next, then you can play around, so you can make a mirror image. And in this way, you're expanding the number of images that is accessible now. You can also rotate it. You can actually change the color of it. And by that, you actually also are able to generate from one, you get up to 500 or multiple hundreds of images. And this is how you actually manage. And then you build a model onto that. In this work, we published very early a study. We were the first in melanoma with AI segmentation. And we have a collaboration with NIH in the Cancer Moonshot program that is the biggest right now. And we have published a number of studies here where we, together with the NIH, have been going forward. I'm very happy. It's a great team and effort. And we were evaluating, and we're just moving into phase 2 of this program. So it's not only US, it's global, worldwide today. Okay, so with this in mind, I show you next study example of where we're going, and this is the MaxMel study. So here you see 4 different type of classes of the melanoma, and the acral melanoma is the most pronounced in Mexico. So this is a study where Diana has taken the lead, and we have 1,000 patients here. And everything I'm showing you now, data, is actually generated in the collaboration done with Covaris technology. So Erik has presented the details of operational of how it's being executed. So here's a workflow that we worked out, going from sectioning, histology, and then going downwards and workup, and then you also need to actually solubilize. So what you want to make a solid sample a liquid, and you want it a fully solubilized liquid. And this has been very efficient and fruitful, and we spent quite some time in getting it right. So, it's always easy to say, yeah, you do like this and do like that. Well, it was more than that. So we were able to get an understanding of the timing, how we should do it, and then, as Erik alluded to, we can do 96 well format in this high throughput system. And this is very important for us because now we are generating study data that you see here. You can really do deep sequencing. You can quantify the 4 different classes of melanoma, and you can also find key regulating proteins in the respective class of melanoma. I will not go into the detail of one of the targets that we are kind of looking into patenting now. But if you want to also have an understanding of how they relate to one another, you can actually do the ACRAL, that is the main importance for us, and then how also you can then do the analysis and check for the other 3. So this is, these are data from 2020, and now we are moving into large scale for the Diana-driven part of the 1,000 patients. And here you see it's actually fresh from the bakery, so we generated these data just before coming here. And this is what you see as a display. So this is the total amount of proteins generated. You kick it up to 6,000, 7,000, 8,000, and then with the database you're able to crunch even further. And when you compute, what you do is then you go in and you need to also to normalize data. So you get an equilibrium, because this is run over a period of time, and you have internal standard, you have controls, and also we have a mixed control that we use. And by doing that, then you can get really reproducible and very strong data too. So you are in the range of around 20% of relative standard deviation of high numbers, which I think is very good at this stage. So here in the end of this presentation, I want to show you some really cool data that we have. So now I showed you on very high numbers, but now if we go to 1 patient, and then we use the protocol with the Covaris technology, and how we haven't, I should say that, we've been struggling a lot we tried actually what is available commercially on the field for solubilization of FFPE study material. Now, it's a wide window scale because in some of the patients you have very, very high stromal content, and the other ones you have not. So it's very heterogeneous too. So you really need a solution where any type of samples can be handled and processed in a good way. So let me show you data on patient, we call him patient alpha. So he's a 56-year-old male. He got colon cancer. That's when we met him, actually, half a year later. And he's still alive. And here is his history in this schematic illustration. So on the top, he got the tumor, and then he has surgical isolation of that. And they were quite many tumors within the colon. And then he proceeded, then he got treatment, then he was tumor-free, then he got recurrence, and then he got prostate cancer, and then another occurrence. So you see, these are 4 events that this patient goes through. So what you need to do in order to be able to come up with some idea of how to solve the problem, you have to do evolutionary studies, and this is an example of that. And I believe that this is the first, actually, that anybody has done on functional level, or proteomics or functional genomics, whatever you want to call it. And what happened here is that the primary first, second, and third metastases were performed in different hospitals. Switzerland, and then in Frankfurt, Germany, and then in Budapest, and then also in London. So we picked all of them up, took us 6 months, and then we moved from there. We did in-depth characterization by pathology, so we knew exactly where we're standing, and we were following the path, and we ran through all of them from the metastasis stages as well. And István, Livia, and Marcel were the pathology drivers here, Marcel from Dublin. So what you find here is a readout where you actually can see that there's a heavy mitochondrial activation that drives the metastasis, and also a very strong push in lowering the immune defense. So the tumor is doing everything to take over. And you will find activated pathways that we were kind of figuring on how best to be able to address in terms of treatment. And also then in the third metastasis, we also saw there's a shift. You know, the primary is not the same disease as in the third metastasis. And here was a TGF-beta activation. So in the end of the day, what we found is we were able to come up with an idea of how to be able to solve. So what you see here is really cool. So the lower part of the red in there, that's a really, really very chewy, very stagnant. It's like actually glycoproteins, and it forms a barrier. It's like a mountain. So that means that immune cells and drugs cannot penetrate. It's like a barrier. That means that the tumor can grow. And that's the way how the system builds in order to be able to say. So anyhow, we came to a conclusion where we could address, and we found a combination of treatment of how you can solubilize these glycoprotein barriers. They can be small, they can be mid-sized, and they can be really large, so they aggregate. So in summary, I would say that we have been able to develop over the last year a workflow, so it will be shortly available in publication. But these are the steps that you do in deparaphrazation. You get cross-linking you have to get rid of, and then homogenization, which is very important. So that is the part that you run it. But also up front, it's very important for us now to get an understanding of morphology. And these are the 4 steps that we do in morphology. So you take a holistic view first, then you segment it, you annotate the cells, and then from there you can actually use your AI-driven models in order to help you to provide directions of where to go. And the 4th step is really to come up with a way forward for optimal treatment of the patient. Very often what we find is combination treatments actually. Okay, so that was it. And of course, the most important is the people. So we have a lot of scientists, some are older, some are younger, fresh blood in, students, postdocs from 8 countries. We like Latino scientists, it's great fun. And we collaborate with many companies also. Covaris is one of them that we really like. So they provided the solution that we needed actually, and we were able to prove that we can move forward. Okay, with that I thank you for your attention.
Question to György. So the question is about the kind of the amount of samples and the processing. So for your study, for the experiment with the Covaris workflow, how many samples at once could you profile with the, with this solution? So how was the workflow organized? That's a really, really impressive, outcome and findings. Beautiful. But how, practically was it implemented?
Well, there's always a first step, right? That means you pay in time and effort, but I think that scaling is not difficult. So right now, we can process a certain number of samples. You work in a 96-well format, so you do, but because you need, in the chain of event, you need to be able to have time to process as well as you go along. You generate the deep sequencing and a turnaround of everything from 8 minutes to 30 minutes in a cycle scale. And then from there, the time-consuming part is really data analysis to crunch high amount of output. That is still today. So here we have developments with computational tools that we are developing that is helpful. And the higher number or the more the database expands, the easier it will be. So we use it.
Thank you.
Perhaps one last addition before we sort of get to the end. So in Lund, the European Cancer Moonshot Center, we work like a hub. So we like to collaborate with companies. We really like the cutting edge developments they have. And we see, and you know, if it doesn't work, we say so. So if you have sort of problems or areas where you feel that it could be helpful, we are there. Please send an email, or if you come by, you're most welcome. We really built a powerhouse in Lund, so you can do a lot. It's really cool. So we like it. We have quite some speed now. So you're welcome.
So thank you, Nadia and György, and all of you coming here. So we end there. Thanks.